this morning and this afternoon I'll be talking about a mixture of things. So for this lecture, we're just going to focus on complex traits. So multifactorial phenotypes, heritability, allele frequency, and also genetic architecture. So just kind of broad brushstroke overviews of these aspects. So multifactorial phenotypes. So as you all are very much aware, many phenotypes are determined by a very large number of factors. Often these are independent of each other, sometimes they're not. And so diseases which demonstrate familial clustering but do not necessarily conform to any recognised pattern of single gene inheritance are termed multifactorial disorders. So these are things that are typically determined by additive effects of many genes, also environmental factors to demonstrate, quite a complex phenotype. So interplay of genes, the environment, and also the interactions between genes and the interactions between genes and environment. And so an important thing to understand is that incidence in close relatives of affected individuals is actually lower than what you would expect if these were caused by a single mutation. So if it's a single mutation in an autosomal recessive or autosomal dominant fashion, you're talking about 25 or 50% risk of a sibling or a child being affected. But for these particular conditions that are multifactorial or complex, these risks are much lower to relatives, but still may demonstrate familial clustering. And of course, as we know, families also share genes, but they also share the environment. There's two general properties when we talk about polygenic traits. So this is resemblance between relatives. So the closer the relationship, the closer the resemblance. And also, I guess this kind of relates to the question yesterday about whether the more pleiotropic, the more polygenic a trait is, the more chance it is of demonstrating pleiotropy. And I guess that is in general true. So you get correlation between traits, so pleiotropy. This next point relates more to animal breeding and selection, that you'll get inbreeding depression, so you'll get a reduction in mean level of fitness, which is restored when you outcross, so increase in genetic diversity. So the metrics concerned with polygenic traits include mean, variance, and covariance. These are no genetic and both non-genetic. And when we talk about the properties of genes, things you want to think about is degree of dominance, manner at which genes at different loci combine their effects, so there's gene-gene interactions, pleiotropy, and linkage and fitness under selection. And then when you start to include non-genetic effects, obviously this becomes extremely complex, and as you're aware, these are simplified by making assumptions. And I certainly recommend that you read Falconer and McKay, which is Introduction to Quantitative Genetics, that goes into this in a lot of detail. So you saw this slide yesterday from David. This is also had involvement from Bei-Ben and also Peter in our lab. And so this is a meta-analysis of all twin studies in the last 50 years, and it's a huge data set, and you guys can utilize this. Over almost 3,000 papers, 17 or close to 18,000 traits and 15 million twin pairs. So it's an extremely good resource to look at. What you can see here, as David pointed out, it's the distribution of MZ and DZ heritability across all of these traits. And by putting all this together, basically what they found is that the average heritability of all these human traits that were measured was close to 50%. And they also looked at the common environmental variants, and they found that to contribute about 17%. So a lot of the heritability can be put down to genetics. And you can actually look up particular traits of interest on this web-based application, which is linked with the paper. And as you can see, there's a lot of different traits in there that utilise what is available in the literature on these twin studies. And so while we can say the average heritability for a particular trait is about 50%, obviously there's a lot of variance. So this is just taking a snapshot of a few of these. This is actually ordered in terms of genetic contributions. And so depending on, so basically even though it's 50%, there is variance within that estimate. So quantitative traits are typically due to the combined action of several genetic loci. And so once you start to get this segregation, you'll get several categories of progeny. And so as the number of loci increases, you get this, the number of phenotypic categories increases, and then the overall distribution approaches normal. So, and this can depend on the cumulative actions of many genes and the environment. I just wanted to briefly talk about epistasis. So I guess it's important to make a distinction between how a variant affects a phenotype from a biological perspective and then how it manifests it statistically. So when we talk about epistasis, it's really defined as the influence of a genotype at one locus and the effect of a mutation or a variation at another locus. However, it's often considered noise in population genetics. So for an example, you've got two variants, so RS1, RS2. So, and biologically speaking, they're strongly epistatic. But if RS2, for example, has a very low minor allele frequency, then within the population, statistically speaking, all you'll see is the linear effect. So complex biological pathways from genetic variants to phenotypes may manifest themselves statistically as seemingly straightforward additive effects. And there is good evidence to say a lot of the genetic effects are working additively, and there's also a paper on this written by Peter Vischer, showing theory and data to support this additive effect. But it doesn't mean the epistatic effect doesn't exist, it's just very hard to measure it in a population sense. So in terms of spectrum of disease and allele frequency and effects, disease associations are often conceptualized into these two dimensions. So here on the y-axis you've got the effect size, here on the x-axis you've got the allele frequency. And I guess the empirical evidence, what we see is that Mendelian disorders tend to be rare with large effect sizes, so they're going to sit up here in the top left corner, and then a lot of GWAS findings are typically associated with common SNPs with small effect sizes. And so the bulk of discovered genetic associations lie in this diagonal, really. And so rare variants with a small effect are actually incredibly hard to detect, and they require extremely large sample sizes for discovery. So this is a GWAS, a recent GWAS on height, and this is actually the power, 80% power curve. So you can see this rapidly increases as you come down to these lower frequency minor alleles. And actually using this 500,000 large cohort, only 83 out of the 700 SNPs were rare variants, and a number of these had quite large effects, which is interesting. So I guess a fraction of the total risk for any complex disease can be due to rare alleles, but it's not expected to be the majority. And this is actually the effect size based on change in height in terms of centimetres. So polygenic inheritance, we can also think of this in terms of a liability threshold. So complex diseases are considered to be threshold dependent. So there's a continuous liability that this is superimposed on. And so the polygenic liability for common disorders is normally distributed. And actually every single person in the population has a differing degree of liability, and everybody carries a unique risk of risk variance. And so any individual on the right of this threshold represents those affected by the disorder. And so given shared genetic and of course the environmental components, the liability is shifted to the left for risk to relatives. So often, I guess people might ask, well, what is the genetic architecture, or what is the heritability of the disease is typically the first question, and then what is the genetic architecture? And so there's four main things related to defining that. So this is really looking at the type and frequency of these risk variants of the complex trait that you're interested in. And so, so how many risk alleles contribute to the disease in the population? And of course this could include multiple alleles within a gene. The frequency in the population of each of these risk alleles, the effect size of each of these risk alleles, and this also encompasses the concepts of penetrance, and so also the way in which risk alleles act together additively or with interaction. So you guys were talking about the ACE model yesterday. This is a paper that looks at different neurological disorders and gives a liability estimate of both the genetic and environmental contributions using this ACE model. So this is from twin and both population studies. And so what they've shown here in this first figure is that most of these disorders demonstrate a large additive genetic component, here in red. And then when they've looked at the genetic architecture underlying these disorders, most of these are associated with common genetic variants, so here in green. And I guess the exception to this particular graph is the severe intellectual disability, and so that's looks like there's a much greater contribution of rare de novo variation. Okay, so summary of this first section is that complex disease can be multifactorial and can include both genetic and environmental effects. Polygenic refers to a genetic architecture of a trait or disease for which many genes can contribute typically in an additive manner, each having a very small effect. The heritability and genetic architecture of traits varies, but is on average 50% and additive in nature. And the liability threshold is the idea that everyone carries risk variance or exposure for a particular disease, and only a small proportion are affected. Yes?
Why is that last point true?
Why is it true? I wouldn't. How?
Like, why do some people go on to develop the disease and some don't?
Well, I guess we'd come back to the point that it's complex, and it could be multifactorial. And so you have a lot of things acting together that might change the risk and push someone over the threshold compared to someone who isn't affected by the disease. Yeah, I mean, it's a difficult question to answer. I mean, if we knew, this is the theory and what we understand about disease. And of course, it's very hard to break that down and say, well, what exactly are those factors? And most diseases we don't actually know, particularly from the environmental aspect. It's environment throughout life, it's infinite. The number of things that you're exposed to, the number of experiences, and this is also acting together with your 50% genetic risk. So I think it's difficult to answer. Okay. So I'll move on to the next section. So this is quite broad. I'm going to brush over geography and genetic structure. We can talk about Hardy-Weinberg equilibrium and how to calculate that, what its applications are. Very briefly about linkage disequilibrium, and then I'm not sure what stage we'll have a break, but later on we'll talk about this different sequencing technology and variation within the genome. So human genetic variation differs throughout the world and is due to the interplay between population movement, admixture, natural selection and genetic drift. Principal components is a very useful and is very commonly used to distinguish between these ancestry groups, and also you can use it as a quality control if you're performing a genome-wide association study and you want to focus on a particular ancestry group. So these two graphs here are two different European populations that are demonstrating a geographic position which corresponds to the principal component separation. So this is just two principal components. It's tilted to kind of reflect the map, and each little dot is an individual, and then you've got the summary of the average of each of those individuals as it relates to the country. And so you can see this actually quite nicely maps the actual geographic location, and then you can also see the same from this part of different European populations here. So this is Tono Escu was involved in this paper, so he might talk about this later. There's other methods to do this. You don't have to use a principal components. There was a recent paper involving Stephen Leslie, and he used linkage disequilibrium structure to map the UK population. So even within a much smaller piece of land, he could actually separate the different groups within the UK or the British Isles, and also the paper actually has a very nice supplementary that talks about how this relates to the history of the UK. So.
How is that different from this principal component approach? I thought the principal components were getting at linkage disequilibrium.
I actually haven't done it, so I can't give you the details. I know that he used a program called Fine Structure, so you kind of step along each haplotype and you say which is the closest relative. So rather than looking at maybe an overall correlation, he's stepping along each haplotype and then pulling in the ones that are closest. So it might be subtle differences in the methods that can actually change the outcome of where they're distributed. So in terms of why is this important, it can have impacts and bias. If, for example, you're collecting cases and controls from, even different cities, that can have an impact on your estimates. So for phenotype, so in this particular paper, they actually simulate some data and use some examples about if you're measuring a phenotype that's modestly correlated with geography, and so they use the example of about 5% of the variance, which is explained by latitude or longitude. They say that the uncorrected p-value distribution shows a clear excess of small values. And so basically, if you don't correct for the variance explained by environment, you bias your result, and you might attribute a larger genetic effect than what otherwise exists. You've heard about the sushi and chopsticks example. This is similarly problematic. So if cases are collected from a separate city to your controls, or you have a different ethnic mix in your cases and controls, then it's difficult to determine whether any association that you find is actually reflecting a population difference in minor allele frequency or is actually associated with case status. So,
That's a good question.
yeah.
Even if we don't look at separate cities, if you have a given population-based data set and you were to do a discovery sample and the other sample, you would split it, you might still even randomly have different proportions of different ethnic groups in both samples. And so how would you correct for that?
I don't know. It's a bit, you should correct for it if you can.
Okay.
So I think it depends on a lot of factors. Yeah, but as long as you recognize that it could be an issue and it might be biasing your result in a particular way, then you should be actively looking at that and critically analyzing your population and the data.
I mean, do people do things like propensity score matching to have two comparable samples, or is that just a concept that's not really applicable to studies like this?
I'm not sure. Propensity matching, I don't know.
Propensity score methods, you need to have some treatment that's like a discrete variable, and it's not really, I don't think that's really applicable here.
Well, no, it seems like, what if you, let's say you control for the 15 principal components and then just do a GWAS regressing genes on city that people live in, then you could kind of discover the things that would be potentially problematic in a lot of different cases.
If you were well powered to do something.
Oh
like
yeah,
that.
you'd have to do this huge size of.
Yeah, I mean, in practice, what's kind of hard about these sorts of things is so when we're doing GWAS, we're combining, like, we don't have the individual level data, and so the things that we're going to try that we can do have to be things that we can get dozens of cohorts individually to do. And so there's a major coordination problem, and especially these people who have the data aren't necessarily, they don't have the technical know-how to do like tricky statistical things. They specialize in gathering this data that they have, and so in general, if you want to do something really fancy, it becomes very complicated in these kind of cohort meta-analysis based type studies. So I think that's been one of the things that really has caused us not to, we just do these simple GWAS, OLS, logistic regressions, because it's, everyone knows how to do that, and it's, despite how hard it is to mess that up, it gets messed up a lot, anyways.
And I take back what I said. I actually think propensity score matching would potentially, you have to think about it more, but it's potentially
Yeah, it seems
applicable,
like it would be fine.
But except for the issue that Patrick mentioned, each of the cohorts would have to do it.
But even if,
Oh no, finish your thought and then I'll ask my question.
I forget what my thought was.
Sorry. I was just gonna ask, is there a growing consensus on how many principal components to control for within a European ancestry population? People always say 10. What is that based on? I've talked to statistical geneticists who say that's over controlling, that you should only at most control for 4 within a European population. And I guess I'm not still clear on why, I don't know.
I think it depends on your data. Yeah, I think you should break it down into your principal components and see how much each component is additively helping, and then define off that. So, it might depend on how large the spread is, but I haven't actually done it, so I don't really want to give you advice, but yeah.
I'm puzzled by the idea that there'd be over-controlling, because if you're finding, it seems like to get GWAS for, say, educational attainment or something, doing 15 or 20 principal components would be unlikely to vitiate that very much. I guess I'm interested in the answer to that, but it seems like the danger of over-controlling is not that high, really.
I mean, that's my sense too, is that given the number of SNPs that we're looking at and things like that, you know, the difference between 4 and 20 in terms of over-control type problems is really limited.
Patrick will actually show you some data on this on Friday from some work we've done with the Health and Retirement Study on the European ancestry individuals, and you'll see there that basically it makes no difference whether you control for 4 or more principal components. I mean, probably because they're estimated noisily once you get beyond the first few, but I agree that over-controlling doesn't seem like much of an issue.
That's what they said, I don't know.
Would it not make a difference in sort of downstream analysis? Like, for example, you ideally want to control just for, population stratification, essentially. And then, for example, if you're doing LDSE and your intercept is below 1 later on, then whatever subsequent analysis you do, interpretation is going to be difficult. But I think if you use LDSE as a guide to sort of say, oh, you've corrected so that the intercept is 1 and there's no sort of inflation in your test scores because of uncorrected population stratification, then that's good. But then if you sort of overcorrect it, it can affect your subsequent analysis.
So I mean, it depends on kind of what stage of the results that we're talking. So there's like the GWAS itself, and you have to worry about stratification there, and so that might affect the weights that you have, and then you also have in principle we're gonna maybe use those results in secondary analyses, and so we might take it into like a replication data set, and that data set may also be stratified, and therefore you need to figure out how to deal with stratification within that data set, and also think about how possible stratification in the discovery sample is affecting things. I'd really be surprised if adding even up to 20 principal components does. I mean, the last principal components might be not doing anything for you, but I don't think it would introduce bias.
So what would it be to over-control? It would mean that you would have to have estimated a principal component that is basically capturing variation in the particular genotype, a lot of the variation in the particular genotype that you're interested in, so that once you control for it, you don't have any variation left, in the genotype to look at the effect of that genotype on the outcome that you're studying. But when you have 2 million SNPs in the data and it's finding patterns of correlation from across the genome, it's very unlikely that anywhere in the first 20 principal components you're going to be picking up a lot of variation in a particular.
And if there is, there's
SNP.
some weird stuff with that SNP. If the 20th principal component is like perfectly defined by a single SNP, you have other problems that you need to be worrying about, I think.
So James, were you going to say something before?
Yeah, just a historical note. There was a paper, I think, in American Journal of Genetics, maybe around 2008, that did propose to use propensity scores constructed from other SNPs in the genome to try to match cases of control. Then some other group came up with another paper saying that method is flawed, and they came up with these simulations showing it doesn't work. And then I suppose you could improve the method, but it doesn't seem to me that it does anything all that much different from principal components or anything else for that matter.
I mean, we've been focusing on SNPs, I guess, because of the GWAS, but let's say there are certain things that are like genetic alleles that are clearly linked to ethnicities, right, which may have an effect on health. For example, think about sickle cell or thalassemia. So I'm wondering, when we do this, are we not losing some of this perhaps important interpopulation differences that may have a huge impact on certain things? Or how do we think about this? I mean, especially because, at least for what I'm interested in, it seems like these are the things that I would find important to focus on.
So it depends on how you use the PCs. So if we just added them as controls, it it shouldn't introduce bias in terms of like the SNP level effects, but it could reduce power, I suppose. If the PCs are explaining some portion of the variation in the SNPs that you're looking at, then you're going to be less powered to see the effect of that SNP on whatever outcome it is that you're interested in. What sometimes we do in practice, just to save computational time, is we first just regress the outcome on a set of PCs, and then we do the GWAS on the residual of that regression. And if in that case, these kinds of things you're talking about could introduce some funny problems for us. But again, you have to believe though that these PCs are explaining a lot more of the variation in the SNP, than I think is reasonable.
But here again, it comes to mind, I'm not fully understanding the link, I guess, between genetic allele and the SNP, right? Let's say you have a gene that only has 2 loci, right? And there's only 3 populations. 2 of the populations have the same 2 SNPs, while the allele that appears is actually the link to the other one, right? Where you have it switched. So if you have basically what's like population level fixed effects, you would get rid of the variation in the SNP, right? In a sense. So that's where I'm kind of.
Yeah. So in the extreme case, so let's take the chopsticks case, and let's say that hypothetically there were an allele that caused chopsticks use, and let's say that in Asian ancestry populations there's always 2 minor alleles at that location, and in non-Asian populations it's always 1. So then there really is a causal effect that you can only identify off of the between group differences. So that's in a case where you would lose all power by controlling for the principal component that eliminates that identifies within Asian and non-Asian populations. So in that case, you could argue, it would be a mistake to control for the principal component because you can't identify the effect. But if the problem is, in that case, that we could never distinguish between the effect of the genotype and the effect of cultural factors that are associated with ancestry. So the approach that we take in these genetic association studies is to make sure we have identification by controlling for ancestry, because if there is a causal effect, we should see it within, if there is any variation, we should see it within group. It might be there's much less variation, so we potentially lose a lot of power, but that's the trade-off we make. So we make sure we have identification and can rule out cultural effects, and then we deal with the power that we have.
I think you could say something a little stronger too, though. If something is only identifiable between groups, that also means it would be identifiable between groups. So you wouldn't need the complicated gene stuff. You just see, okay, this group is different than that group, and then you have the big debate, okay, is that because of culture or is that because of genes? But whatever evidence there was between the groups would be evidence that people would have already seen without going through any of the gene work. It's going to be very equivocal evidence, but what evidence there is already there.
So I think the professors have kind of answered my question just now, but I'm wondering, are we almost always agnostic about what the principal components are related to? For example, the PC1, the principal component that controls or explains the most variance is the geographical factor. But if, for example, we have included 10 more principal components that we don't know what those principal components are explaining, right? So in the equation, if we want to regress the phenotypes on a set of principal components and want to control for the cultural factors, that would be over control, but we don't want to do that, right? So essentially we're trading off knowing what we're controlling for versus controlling as much as possible.
So I feel like the use of principal components to deal with stuff like this is a little bit ad hoc. Like we do it because we know it works, or because it appears to work. And so when we we see that if you don't use principal components, you get all these signals, and then by adding them, it behaves in a way that's consistent with kind of what we're worried about. And then we also can look at the principal components, and it correlates, and they tend to correlate strongly with the things that we think are causing problems. And so like in European samples, the first two principal components tend to be like latitude and longitude. And in American samples, one of the first 10 principal components is often highly correlated with Hispanic. It's not that we actually know what these are, but we just know what they tend to be, and it seems to be effective. And so, I don't think we should think of them as like these magical things. And something that we're worried about now is that we know that they're pretty good at absorbing kind of the largest biases that get introduced by stratification. It's not totally clear that it's picking up subtle stratification. So there might be a little bit of stratification that's left when we're done. And when we're finding the largest genetic effects, maybe that's fine. As we start getting these really large sample sizes, the amount of stratification that we're not able to capture with principal components may be comparable in magnitude to the effect sizes that we're seeing now. And so it's, again, I don't think we should think of these PCs as solving the problem, but it's something that just kind of an ad hoc thing that seems to work.
So we can keep talking about principal components now, and it's a good discussion, but we are going to be talking about them in more detail in a couple of days and tomorrow and Friday, I think.
Oh, okay, yeah, that's true.
So, so just keep that in mind, that, the detailed, might be better to save detailed questions for later and let,
Alright, so if you're going to address this tomorrow or whatever, then just tell me, but I was wondering how you guys pick how many PCs to control in general. Like,
let's defer that.
Alright.
Let's defer that. But ask it again.
Sure.
All right, so one other process that we typically use in GWAS QC, and I think there's also going to be another lecture about this, but I'll introduce Hardy-Weinberg equilibrium. So this particular theorem states that in a large randomly mating population with no selection, no mutations, migration, genotype frequencies should generally stay constant from generation to generation. So basically, in here, with this, you're assuming conditions of equal fertility, equal gene frequencies in both males and females, and equal viability. So absence of any evolutionary influences. And so if you show a deviation from this expected Hardy-Weinberg equilibrium, it's suggesting that your conditions have not been fulfilled. And in a practical sense, this can be, you might have related individuals in your sample, and often, or more often than not, there might be technical errors. So you might have a non-specific SNP, you might have poor coverage specific to a particular genotype, so you might be losing all your homozygotes and only counting the heterozygotes, for example, and that can really change your result. So by testing for this Hardy-Weinberg equilibrium, you can take out these SNPs that aren't really capturing something that you're interested in. So I just have here some quite old data, but it's blood groups from Greenland and Iceland. These are your genotype frequencies here. So A1 is the homozygote, A2 is the other homozygote, and you have the heterozygotes here. The proportions are quite different between these two countries. And then also you have the allele breakdowns, and these are the number of individuals. So I don't know if any of you have been exposed to this, but I can show you how you can actually address this question, so whether these genotype frequencies are actually in Hardy-Weinberg equilibrium. So there are 3 possible genotypes, as I've just shown you, in this particular population. So if we just look at the Iceland numbers, if the population is in equilibrium the gene frequency should be the same in the parents as it is in the progeny. So the gene frequency, so let's assume this is the progeny, so the offspring. And then we can use these frequencies to test the parental frequency, and then you can kind of calculate the genotype frequencies that you would expect based on Hardy-Weinberg. So we're not going to derive this, but the genes from the parents we know either are going to be an A1 or an A2. And here we actually have the genotypes in the progeny, and so all we do is just work out the gene frequency, or the allele frequency I should say. So the way you do this is a homozygote, so it's just 233 times 2, and then plus also have a set of A1 alleles here, so you plus 385 over the proportion of total alleles, so every single person has 2 alleles, so it's 747 times 2. And then from this number, as a proportion, it's actually 0.57. And then we know both alleles add up to 1, so you can either go 1 minus 0.57, or you can calculate in reverse the A2. So if we're calculating how many A2 alleles, it's 129 times 2, and then plus 385 over a proportion of this, double this number. So this is basically what the parental allele frequency would be. And then we use this, we use these alleles here, and this is the Hardy-Weinberg equilibrium equation, which basically says the two A1s is equal to P squared, this being P, this being Q, and the other homozygotes are also equivalent to Q squared, and the heterozygotes equivalent to 2PQ. So if you substituted in these numbers here in this equation, you would come out with an expected homozygote, heterozygote, homozygote genotype frequency. And then you just use a simple chi-square test to see if this deviates from what you would expect. Your observed numbers deviate from what you expect. So I don't know if any of you have calculated that on the go, but this is what you should have. This would be your chi-squared, and the p-value is 0.2. So you would say that this population is in Hardy-Weinberg equilibrium and is close to what you might expect it to be, given this a randomly mating population. Okay, so we briefly mentioned this yesterday, and also there's going to be more about this in Raymond's lectures. But linkage disequilibrium in populations, so as you're aware, autosomal material on chromosomes is inherited sometimes in regions, and these regions can remain the same over generations. So this being the ancestral chromosome, and here you say that here you can see present day chromosomes, and this region has remained the same. So alleles in, so linkage disequilibrium, what is the definition? So this is a non-random association of alleles at 2 or more loci that descend from a single ancestral chromosome. So if your alleles are in linkage disequilibrium, they're inherited together more often than you would expect by chance. So correlations among neighbouring alleles reflect haplotypes that descended from a single ancestral chromosome. And what is a haplotype? So this is a consecutive sequence of alleles that are at different loci on a single chromosome in an individual. So due to many reasons, so migration, mutation, selection, a small finite population size like a bottleneck or other genetic event that the population experiences may be why a particular haplotype exists. And this, of course, is broken down by recombination. Yes.
This is still about the Hardy-Weinberg equilibrium. So,
Yep.
I mean, what level should you set? Because if you set a 5% p-value threshold, then there'd be 5% of the genes that might be okay that you'd be kind of throwing out because they didn't look in Hardy-Weinberg equilibrium. So is the practice to just say, okay, that's okay if we lose 5% of the genes that genuinely are okay?
So I think Aisu Akbe is going to talk about this tomorrow, but David, do you want to give a quick answer? Oh. Okay, let's wait till he comes back. He's the expert on this.
Yeah, but there is cases where it deviates by chance, of course, and there's lots of papers that you can find and say, well, that population isn't even in Hardy-Weinberg equilibrium. So, yeah, I mean, depends where you want to draw a line. I think typically when you're doing a GWAS, you have so many SNPs that if there's any chance there could be what you would expect to be a technical error, then you'd throw it out. Yeah. But we'll wait for David. Okay, so what can linkage disequilibrium in populations be used for, and how is it used in GWAS? So it can be exploited for a few different purposes. So it can give you an understanding of the history of our ancestor populations. SNP panels can actually allow the estimation of the expected genetic covariance between distant relatives, so unrelated. Genetic variants of a disease or phenotype captured by common SNPs can be assigned to chromosomes and chromosome segments. And then the total genetic variance due to linkage disequilibrium between common SNPs and unknown causal variants can be estimated. And this is just a picture of different haplotype blocks within a chromosome.
So is this done in interaction with the first couple principal components? Because you'd expect linkage disequilibrium to be different for different geographical groups, or is it just done for all of Western European linkage disequilibrium together?
This is just saying that you can use linkage disequilibrium for different purposes. So if you want to look at the history of an ancestor population, then your processes would be different to what you would do if you're looking at genetic variants. Yeah. So another main reason that linkage disequilibrium is actually useful, and the sharing of haplotypes essentially, is that it's widely used for imputation in GWAS. So the reason that you do this is that you can improve the resolution of a marker trait association and map it to a smaller section of the chromosome. So this is an example of what happens on the SNP array. You are not looking at, while you look at GWAS is genome wide, but you're not actually looking at every single base in the genome. You're looking at interspaced bases. And so you kind of you have, these gaps in your data. And so there's actually some deeply sequenced reference haplotypes. So this is it was originally the HapMap project, and this is now been taken over by the 1,000 genomes, or it's more than 1,000 genomes, but these are very densely genotyped populations that do have these reference haplotypes for you to be able to use this data. So basically, you can fill in the gaps between your SNPs that you've actually measured in your population. So the performance of this imputation and GWAS studies really depends on the local distribution of the linkage disequilibrium between the nearby variants. And so I think the most recent correlation accuracy between imputated and experimental genotypes for common variants was over 95%. And then this, this correlation accuracy drops gradually with minor allele frequency. So the next few slides, yes?
How sensitive is the linkage, equilibrium to essentially the population that I'm using? So if I'm using LD in a European population, can I easily extract, like, do the same procedure in very different population, like an ethnically different population?
Yes. Yeah, you can. And actually, it's the accuracy, I think, for the imputation is better in Africans than it is in Europeans because the haplotype blocks are shorter. So the imputation is better.
But
the way I understand it.
The reference.
Reference.
Are from Sigma population, right?
So the reference haplotypes are from the 1,000 genomes, which is actually 2,500. And that's from distinct populations that have been densely genotyped. And that includes a European population. Yes, David?
So in the early days of imputation, there was some uncertainty about this, and some people thought you might get more accuracy by just imputing against a reference population, and that's somehow similar in terms of ancestry to your sample. And by now, everyone just uses all the available data, it just gives you more haplotypes, and empirically seems to be better that way.
David, there was a question that came up before while you were out of the room. Miles asked about what p-value threshold you use for testing for Hardy-Weinberg equilibrium. And so, you know, I said Aisu's going to address that tomorrow, but you could give a quick answer now.
Yeah, so the answer is that it's a little bit odd to use a single p-value threshold because it's sample size dependent, right? So why might you get departures from Hardy-Weinberg? So first of all, why do you drop SNPs that fail Hardy-Weinberg in QC? The reason is that if you think you see really radical departures, something went wrong with the genotyping or the imputation, and so you just don't trust those SNPs, and you want to get rid of them. So if you have a sample with millions of individuals, even a tiny violation that could be driven by something like assortative mating might cause you to reject if you have a p-value threshold of 10 to the negative 5. 10 to the negative 5 would mean something very different than 2,000 people. So in practice, I think you want to use some sort of notion of distance from some metric of distance from the expected frequencies and not the p-value, but that's not what people do. You kind of, I think you arrive at, through a process of trial and error, to some p-value threshold that applies to your specific sample, if that makes sense. And this is something that people, when they see this the first time, they often have, they often find it confusing to drop SNPs that are in Hardy-Weinberg because they think, well, you know, maybe the SNP is done with their selection, or maybe you have, but maybe there's a sort of mating, maybe there's something real and interesting about the SNP that causes the violations, and that's not why they're being dropped. They're being dropped because the violations that you're seeing are much more dramatic than that.
Can I ask again about the imputation? Because you say you're using all the data, but how is this working? Let's say I take my genome and now I get the chip and I want to impute. I just try to find with which of the many populations that you have the haplotype data that I link better and then from there impute, or how is this working exactly?
I'm not sure I understood the question.
Because I imagine that, let's say that the imputation depending on my ethnic background will be different, and so you only have 26 populations in the 1,000 genome data,
yes.
right? So, well, first I don't know if this will vary a lot. Let's say if I take one ethnic group in the middle of the sub-Saharan Africa and I look at different ethnic groups close by, if that will change a lot or not. But then even if I don't know my ethnicity then, or like in the sense that's what we have here, because you won't know, how do you go from there to figure out who you should be using, and how do you know how noisy that would be?
So in practice, what you do is that you, in practice these days, what people do is they use all the available data. Most of those data are publicly available, so you can actually do it online on secure servers, and then there's algorithms to work it out for you. And the how do I know how accurate it is? I think intuitively, the accuracy is going to depend on the extent to which you have, if you have a lot of haplotypes in your genome that aren't in any of these populations, so that then imputation accuracy is not going to be as good, all else equal. But that should be reflected in the imputation uncertainty statistics that are returned in your computed data. Right? So using this information about LD, they can sort of assign the probability that given that I have these SNPs for you, that that this is your actual haplotype.
I'm just thinking, I don't know whether we should stop now and then.
Well, you can go for, they're scheduled for another 10 minutes, but
Okay.
I think now's a good time. We could change the.
Okay, I'll do these technology slides. Okay, so I think I just have two of these, just to summarize, what is the sequencing technology? How does that actually work? What is being used now? What is coming up? So Sanger sequencing was developed in the late 1970s. This is actually still being used today. So it essentially works by using a chain termination method. So you have single-stranded DNA, and this has, you have two primers going in opposite directions. A DNA polymerase copies the DNA by adding nucleotides to a growing chain. And then there's also some dideoxynucleotides which lack a hydroxyl group, and this results in a chain termination at either A, C, G or T. And so what you'll recover is a mixture of different sized DNA fragments that are obtained, and so if you run it on a gel or you pass it by a laser, it allows base calls to be made. And so this method is actually quite accurate, but it isn't as efficient as the newer methods, and it has quite a high cost per run because of this reduced efficiency. So typically the way you'll see a result is that this is a chromatogram, and these are the bases at each position that it's calling. And you can see these are coloured by different fluorescent. So this has been run through a laser. So one of the most common methodologies used since then is a SNP array. So this was developed in the late 90s. It obviously included much fewer SNPs than what we have now on the arrays. And so I think around 2005 was when we started to get up into the 400,000 to 500,000 variants per chip. So this uses a hybridization approach. So basically your input is fragmented DNA, so short fragments which bind to a complementary probe. So the probe being 25 to 50 base pairs per site, and you'll have multiple probes at each site, and these contain a fluorescent signal. So the colour and the intensity of the signal represents the number of matched probes, and this can determine the genotype and allow you to call homozygotes from heterozygotes at each of those probes. So second generation or next generation sequencing was developed around 2005. So this is high throughput, massively parallel, short read, next generation sequencing. From my research, basically there are quite a few different platforms and different methodologies. And what this can do is measure both exomes, so just the protein coding regions, and sometimes a little bit of overlap between each exon, but also you can look at the whole genome. So this is every single base pair. So this is a huge amount of data that you can get from this sequencing approach. Illumina uses something called sequencing by synthesis. So what happens here is that they immobilize many different fragments on a plate or a flow cell, and they sequence runs simultaneously on these fragments. They use something called a fold-back PCR, and so I put in a link there that actually explains how this works. But basically, this amplifies the fragmented DNA into a cluster, and tag nucleotides are incorporated into the strand of the DNA, and the base calls are made at each addition. So these fragments are then aligned to a reference, and you of course have, as this picture here shows, this is some data from exome sequencing, but basically you'll have different coverage at each base that can allow you to make an accuracy call on how confident you are that that is the base. So this is an extremely cost-effective, it's high speed, and it's quite accurate. So the third generation sequencing, so this is starting to become more used now. This was developed in 2003 and the technology has improved. Were you going to ask a question, Megan? Okay. So this includes, so it's also called smart sequencing. So this stands for real-time. It's like a real-time platform, and you've heard of PacBio, which also is Pacific Biosciences, and I think this is the most widely used technology in this third generation realm. So DNA polymerization occurs in arrays of nanostructures. So these are called zero-mode waveguides, or ZMWs. And these are actually very, very tiny holes in a metallic film, which cover a chip. So the DNA, or the library of interest, washes over, and the fluorescent DNTPase, which is extends the DNA chains by single nucleotides, are monitored in real time, and it displays like a detectable fluorescence during the incorporation, after which the dye is cleaved away. And so the beauty of this method is that it's capable of very long reads. So much greater than, for example, 10 kb. So this is extremely useful for de novo genome assemblies. But I think the drawback in this method at the moment is that it's got quite a high base error rate. So just to show that, this is observed error rate. And you can see the PacBio is actually quite a lot higher than the other next generation sequencing platforms. So there's limitations to a lot of these sequencing approaches. Some regions in the genome are difficult to resolve and will not be accounted for due to insufficient read lengths. And you might get unresolved structural variation. There's also sequence content and bias. So particularly in areas where you have a very high GC content, or if there's a lot of low complexity. So lots of dinucleotides all in a row, or trinucleotides, it can have issues mapping that back to the reference genome.
What's a dinucleotide?
Just two bases repeated. So just like CG, 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 just, or trinucleotide, same thing with an extra letter. And so also structural variations, so inversions, insertions, deletions, that's not captured well by this next generation sequencing. Simple and complex repeats, so that includes microsatellites. Also telomeres are actually quite repetitive sequences, so this isn't covered well. And anything that's very highly polymorphic, so like your HLA region. So, I mean, the costs are different, but this is changing all the time. So basically, when it comes to the time of choosing a sequencing method, yeah, that's when you should look at the costs.
So how does the cost compare for getting every base versus just getting these samples along the way? Right now?
It depends how you calculate your cost. Because, I mean, the initial output, maybe it might not differ all that much. You know, maybe it's maybe it's $1,500 to do a whole genome, and a SNP array maybe closer to be closer in the hundreds, but you get a lot more data with your whole genome. But then you've got to process it, and so and as we know, QC is 99% of the workload in these genetic studies. So depending on how familiar you are with the data, what kind of data storage you have, what are the costs of that to you in terms of time and efficiency, I think it can really vary.
So
Yeah.
the kind of estimates I've heard on cost, at least for the kind of data that we deal with, is that sequencing would cost something like $500 a person, and genome-wide genotyping is like $50 a person these days, $40 to $50 a person. But
Okay.
Fleur says you have to do a lot more work with the sequencing data. And
Yeah, so that's just to get the raw data from your, so you've already extracted the DNA, you put it into plates.
Exactly, that's the cost of the actual getting the
data. Yep.
But then, as you say, you have to have much more powerful computers to deal with sequencing data, and we don't really know as much about how to do the QC on them, and it's messier. Well, could you say maybe just a little bit about what are inversions, insertions, deletions, microsatellites, and VNTRs? Just because we're going to be talking almost entirely about SNPs, but it's good for everyone to know at least what these other kinds of variants
I'm going
are.
to talk about some structural
You'll talk about them soon.
variations. Yeah, I mean, inversions is just pretty much when a base might flip over. Then insertions is just an extra base, in a sequence, or and the same thing with a deletion. So it could be one base, it could be 5, it could be 10, and so this can impact on, if it's just one codon, then it's okay. But if it's a single base, and that means you'll get an entire frame shift, so that can be more severe. And these structural variations are difficult to pick up in these fragmented next generation sequencing approaches.
But even if you don't actually measure them directly, sometimes you can tag them. They can be correlated with SNPs, right? So then
Sometimes, yes.
Yeah. So sometimes you can detect them even though you don't measure them well.
Yep. Yeah, I'll show you. So yeah, you can tag them, but the other thing to know is that things like microsatellites are actually, these are things that you use in forensics, and these are highly different between each person, even within families. So these are highly mutable sites. So when we talk about tagging, we're talking about common sites, but then, yeah, I'd say sometimes you could tag it, but there's a lot of changes between individuals. So.